Good afternoon, grade 10. Today, our lesson is about cell division. This is teacher Mildred, and this is biology, grade 10. To start with, let us first describe chromosomes. What are chromosomes? In the nucleus of every cell, there are a number of long threads called chromosomes. Most of the time, the chromosomes are too thin to be seen except with an electron microscope. But when a cell is dividing, they get shorter and fatter, so they can be seen with a light microscope. Each chromosome contains one very long molecule of DNA. DNA stands for deoxyribonucleic acid. The DNA molecule carries a code that instructs the cell about which kinds of proteins it should make. Each chromosome carries instructions for making many different proteins. A part of a DNA molecule coding for one protein is called a gene. It is the genes on your chromosomes which determine all sorts of things about you. For example, what color your eyes or hair are, whether you have a snub or rounded nose or a straight one, and whether you have a genetic disease such as cystic fibrosis. You inherited these genes from your parents. Each species of organism has its own number and variety of genes. This is what makes their body chemistry, their appearance, and their behavior different from those of other organisms. This explains why human beings are different from plants and from other animals. Humans have a large number of genes. You have 46 chromosomes inside each of your cells, all with many genes on them. Every cell in your body has an exact copy of all your genes. But unless you are an identical twin, there is no one else in the world with exactly the same combination of genes that you have. Your genes make you unique. Let us now study cell division. You began your life as a single cell, a zygote, formed by the fusion of an egg cell and a sperm cell. The nuclei of each of these gametes contained a single complete set of 23 chromosomes. When they fuse together, they produce a zygote with 46 chromosomes. A cell with a single set of chromosomes such as a gamete is said to be haploid. The nucleus of the zygote contains two sets of chromosomes. Therefore, it is a diploid cell. The chromosomes in a cell of a man and a woman are different. The photo shows the chromosomes in a cell of a man and of a woman. They have been arranged in order, largest first. You can see that there are two chromosomes of each kind because they are from diploid cells. In each pair, one is from the person's mother and the other from their father. The two chromosomes of a pair are called homologous chromosomes. There are two types of cell division, mitosis and meiosis. Mitosis. Soon after the zygote was formed, it began to divide over and over again, producing a ball of cells that eventually grew into you. Each time a cell divided, the two new cells produced were provided with a perfect copy of the two sets of chromosomes in the original zygote. The new cells produced were all genetically identical. This type of cell division, which produces genetically identical cells, is called mitosis. Just before mitosis takes place, the chromosomes in the parent cell are copied. Each copy remains attached to the original one, so each chromosome is made up of two identical threads joined together. The two threads joined together are called chromatids, and the point where they are held together is called the centromer. Two new cells are formed after mitosis, each with one copy of each of the four chromosomes. As the new cells grow, they make new copies of each chromosome, ready to divide again. Mitosis is the way in which any cell, whether it's a plant or animal, divides when an organism is growing or repairing a damaged part of its body. It produces new cells to replace damaged ones. For example, if you cut yourself, new skin cells will be made by mitosis to help to heal this wound. Mitosis is also used in asexual reproduction. You have seen, for example, how a potato plant can reproduce by growing stem tubers which eventually produce new plants. All the cells in the new tubers are produced by mitosis, so they are all genetically identical. The second type of cell division is meiosis. Meiosis is a reduction division in which chromosome number is halved from diploid to haploid, resulting in genetically different cells. We learned a while ago that gametes have only half the number of chromosomes of a normal body cell. They have one set of chromosomes instead of two. This is so that when they fuse together, the zygote formed has two sets. Meiosis is a nuclear division giving rise to cells that are genetically different. Human gametes are formed by the division of cells in the ovaries and testes. The cells divide by a special type of cell division called meiosis. Meiosis shares out the chromosomes so that each new cell gets just one of each type. You may remember that one of each pair of homologous chromosomes came from the person's mother and one from their father. During meiosis, the new cells get a mixture of this. So, a sperm cell could contain a chromosome 1 from the man's father and a chromosome 2 from his mother, and so on. There are all sorts of different possible combinations. This is one of the reasons why gametes are genetically different from the parent cell. Meiosis produces genetic variation. The photo shows the difference between mitosis and meiosis. From the parental cell, there is replication, cell division, and after cell division in mitosis, you produce two identical daughter cells, while in meiosis you form four daughter cells which are unidentical to each other. Here is another illustration that shows the difference between mitosis and meiosis. Again, after the replication of DNA, in mitosis you produce two genetically identical daughter cells, while in meiosis you produce four daughter cells which are genetically unidentical to each other. Stem cells. Shortly after a zygote is formed, it begins to divide by mitosis. Over the next few hours and days, the cells divide over and over again. Each division is done by mitosis, so every new cell is genetically identical. However, as the embryo develops, the cells begin to take on different roles. This is called differentiation. Some of them will become skin cells, others muscle cells, others goblet cells or white blood cells. How does this happen? Every cell in your body has the same genes, but in each cell, only a particular set of these genes is switched on or expressed. The cells in your hair follicles, for example, are the only ones that actually express the gene for hair color. This gene is present in all your other cells, but it is not expressed. So differentiation involves switching on particular sets of genes or expressing a particular set of genes on or off. The cells in the very early embryo, before they start to become different kinds of cells, are called embryonic stem cells. Embryonic stem cells are able to produce every kind of specialized cell in the body. But once a cell has differentiated into a particular type of cell, then it cannot change its role. A muscle cell, for example, cannot divide and produce liver cells or skin cells. By the time you have become an adult, most cells have differentiated, but some stem cells still remain. They are called adult stem cells. Like embryonic stem cells, these are able to divide to produce different types of specialized cells. But the range of different cells they can produce is limited. For example, you have stem cells in your bone marrow that can divide to produce red blood cells, platelets, and the different types of white blood cell. But they cannot produce nerve cells, liver cells, or any other kind of specialized cells. Medical researchers are very interested in finding out more about stem cells. It is likely that we will be able to use them to help to cure diseases that are caused by some of our cells failing to work properly. For example, we all know that diabetes 1 is caused by the loss of the pancreatic cells that secrete insulin. If we could use stem cells to replace this, then it might be possible to cure this kind of diabetes. And that's the end of the lesson about cell division.